Genetic diseases are becoming more prevalent in today's society. Scientists are constantly looking to increase accuracy and find alternative, more suitable models to represent pathways of diseases. Although not a recent discovery, scientists have been using fruit flies, Drosophila, as models for human genetic disease for almost 30 years. It is only with new genomic tools such as UAS-Gal4, which allows for quick analysis of human disease-associated variants for function and pathogenicity in flies. Proving to be extremely difficult to model in mammals, Drosophila proved to be a relatively easy and affordable model. This genomic tool will be covered in more detail, followed by an example focusing on Alzheimer's disease, followed by advantages and disadvantages of using Drosophila as a model for human genetic disease. Before delving into how genomic tools work in a fruit fly, we need to give some brief background information of Drosophila. The Drosophila melanogaster model organism has proven to and continues to prove to be an extremely ideal model for looking at genetic disease. One of the advantages being its short life cycle. The female fruit fly, approximately 3 mm in length, will lay on average 40 to 50 eggs a day. That is a lot. The life cycle of the fruit fly only takes about 12 days to complete at room temperature. After the egg is fertilized, the embryo emerges in less than 24 hours. The embryo undergoes successive molts, which is shedding of the outer skin, to become the first, second, and third insta, intermediate development larva. This is followed by the pupal stage and finally the emergence of an adult fly. Because the flies themselves are quite small, it is incredibly easy to raise a lot of them at once. Genetic experiments can be done in a shell vial with just a few flies, therefore many different mutant stocks can be maintained and numerous experiments carried out at one time in a small lab space. As we know, genetic information, DNA, in all cells is carried in chromosomes. The low number of chromosomes in Drosophila, 4, is key to this organism's success in genetic modelling. The reduced genome complexity allows easier interpretation. The full genome sequence of Drosophila was published in 2000. The fly genome is only about 5% of the size of the human genome, that is, 132 million base pairs for the fly and 3.2 million base pairs for the human. The fly has approximately 15,500 genes on its 4 chromosomes, whereas humans have about 22,000 genes among all their 23 chromosomes. Humans and flies have retained the same genes from their common ancestor, over about 60% of their genome. Therefore, Drosophila is extremely good to be compared to the human genome. Now we will move on to how the genetic tools work with an example of a genetic disease. Due to the fly's relatively short lifespan, complex questions of brain function can be addressed more rapidly than other mammal models. Although the gross anatomy of Drosophila and the human brain are very different, they share numerous genetic, cellular, and chemical properties. Alzheimer's disease is the leading cause of dementia and the most common neurodegenerative disorder categorized by the shrinkage of several regions of the brain, explaining early symptoms of memory loss and cognition. The buildup of amyloid beta peptides is a sensation found explicitly in AD development. This protein accumulation is proven to be toxic in the brain and thus causing AD. To turn Drosophila into an AD model, recent studies have employed the use of UAS-Gal4 system, which ultimately can turn genes on or off. So let's break this down. We have 2 lines of flies, one line being the driver line and the other line being the responder line. The driver line contains a Gal4 coding under a tissue-specific promoter and drives expression of the protein of interest. The responder line, made up of upstream associated sequences, UAS, and the human gene associated with AD, will respond to the driver and express the gene. The driver line and the responder line sequences are not expressed individually, they have to be fused to be expressed. Gal4 is the transcriptor factor and will sit on the UAS of the responder line and drive the expression when crossed together. The result is the expression of a target gene, in this case simulating Alzheimer's disease in the experimental fly group. The control group will contain the same strain of Drosophila flies, but without the inserted protein associated with AD. The behaviour is then assessed, and it was concluded that flies with the associated protein showed characteristic differences and brain efficiency decline compared to the control strain. This is all occurring in the control nervous system of the fruit flies. Advantages of using Drosophila as a model organism include no ethical problems, easy and cheap to maintain, fast and inexpensive, short generation time, and the fully sequenced and annotated genome. The disadvantages include brain anatomy and cardiovascular system differing substantially to humans, no easy measure of complex behaviour, only basic measurements of cognitive decline, and less complex. Overall, Drosophila models have successfully provided valuable information on AD. Furthermore, due to its simple brain anatomy, Drosophila is ideal for in vivo testing, but it's anticipated that, as techniques and analysis progress, Drosophila models will assist in the development of disease treatment and ultimately a cure for Alzheimer's.